Hello students, I welcome you to EPG Pathsala. I am Dr. J. Arunachalam from Sastra University. Today we are going to talk on a module on a tutorial on basic BioPython modules for handling sequence and structure data from the paper Bioinformatics. The objectives of this tutorial is we have to introduce BioPython methods for handling macromolecular sequences, which includes protein sequences, DNA sequences, and how to parse them, manipulate them, and how to access these sequences through BioPython using from various databases, as well as how to handle various file formats by using BioPython. So the objectives are parsing sequence data from various databases and file formats, parsing, handling, manipulating sequence alignments, parsing and analyzing macromolecular structure data as well. BioPython is a collection of freely available Python tools for bioinformatics and computational biology. BioPython has many useful features like parsers for various types of bioinformatic files and data, and it also helps us to access online data as well as other online services. Like for example, if I want to use sequence alignment programs or some phylogeny tree creation programs, I can easily access online through my code itself. I don't need any graphical user interface to do the purpose. Suppose if I have a sequence and I want to perform a local alignment on that sequence against some database, I can what I can do is I can write a BioPython code which can automatically search through the internet, access the database, and do the local alignment search. So this becomes very simpler using BioPython. When I have just a single sequence, it is easy. For example, if I am performing a blast search, when I have just a single sequence, it is just easy to do it on web-based interfaces. But when I have thousands of sequences, how am I going to do that on web-based interfaces? It becomes difficult. I can't keep on clicking 1,000 times. So it would be very useful if I write that in my code, in my Python code using BioPython modules. I can easily do that as a job which runs on background in my computer. So BioPython also has interfaces to many important and frequently used bioinformatics programs like BLAST, ClustalW, DSSP, etc. BioPython and its dependencies can be obtained from the URL http biopython.org by key download. BioPython can be downloaded from there. It is completely free and it could be installed in your computer. BioPython runs on many platforms like Windows, Mac, various flavors of Linux and Unix, and you just have to download it and install it along with your the regular Python installation. So more detailed instructions for installations can be obtained from the URL biopython.org dist docs install as a PDF file, or if you want to read it as a HTML file, it is also possible. Sequence objects and sequence record objects. BioPython has two different methods for handling sequences. So it has a sequence object and it has a sequence record object as well. Sequence objects are much simpler, whereas sequence record objects have some additional features which is not present in sequence objects. So sequence objects, additional attributes that is present in sequence objects differentiates them from simply handling sequences as strings. For example, consider a sequence AGTACA CTGT which is displayed. We can create a sequence object with this particular sequence as follows. So BioPython, the Python prompt appears, after which you will have to type the command. So as a first step, you will always have to import the corresponding modules that I want to use in my program. So here I want to import bio.seq from the seq module. So I have to say from bio.seq import seq, and then I say my underscore seq, which is the name which I have for the sequence object that I want to create. If you, it is a name that we select. So I have selected it as my underscore seek. So I say my underscore seek is equal to seq seek with s caps. Python is case sensitive, so it is important that s should be caps. That seq seek is the function that creates the sequence object. So I have to say seek, and then in brackets I will have to give the sequence within the quotes, because it is a string. So if I want to display that, I just have to type the name of the sequence object that I have created. I just have to say my underscore seek, and the sequence object will be displayed. Sequence objects and sequence record objects. MySeq is the name of the sequence object we have created already. It has an additional attribute called alphabet. In this case, it is empty. So our alphabet attribute, which we already created in MySeq, is empty, but it has an attribute which is denoted by empty braces. The accepted attributes for a sequence objects are IUPAC protein, extended IUPAC protein, IUPAC unambiguous DNA, IUPAC ambiguous DNA, extended IUPAC DNA, IUPAC unambiguous RNA, IUPAC ambiguous RNA. So these are the list of accepted attributes for any sequence object. So in IUPAC protein, we have standard amino acids. In extended IUPAC protein, we have some non-conventional amino acids which are present in sequence. For example, protein sequences which are derived from structures contains amino acids like selenocysteine, which are not part of the conventional 20 amino acids that are present in the protein. This selenocysteine is incorporated to obtain the phases for solving X-ray crystallographic structure. Nevertheless, in that structure, that sequence will contain a selenocysteine, and we need a separate attribute to describe such types of sequence objects. So we have selenocysteine. Sometimes in protein sequences, we have X, which describes that there is an amino acid there, but there is some ambiguity in determining what amino acid that is exactly. There are several other examples like hydroxyproline and other amino acids which are not part of the conventional 20 amino acids. If our sequence has such type of amino acids, we bring them inside extended IUPAC protein. So the attributes for such type of sequences is extended IUPAC protein. Similarly, for DNA sequences, unambiguous DNA sequences, we have the conventional GATC letters which are normally present in any DNA sequence. In ambiguous DNA sequences, once again we have non-conventional bases, and we can also have bases which are denoted by N, which says that it is similar to X, what we said in protein. It says that there is a base there, but we are not able to determine, the experiment is not able to determine what base exactly it is. So it is denoted by N. Such type of sequences should come under ambiguous DNA, and the corresponding attribute for them in sequence object is IUPAC ambiguous DNA. Same could be done for IUPAC cDNA, which has modified bases, and the same could be done for RNA sequences as well. As unambiguous RNA contains G, A, U and C, but ambiguous RNA might contain pseudouracil or other modified bases which are present in RNA sequences. Sequence objects. Sequence objects with alphabet information can be created in BioPython as follows. First, you have to import the bio module. So I have to say import bio, and then from bio.seq import seq. So that is the module that helps us to parse as well as manipulate sequences in BioPython. So you will have to say from bio.seq import seq, and then the next line we have to mention from bio.alphabet import IUPAC. Since we are creating sequence objects here, in the previous slide we saw various IUPAC ambiguous and unambiguous alphabets that are associated with the sequence object. So we will have to import that module as well. So from bio.alphabet import IUPAC, and then we can create the sequence object as follows. MySeq, I have named the sequence object as my underscore seq here, but it could be anything. That is the name that we define. So here I have named it as my underscore seek. My seek is equal to seq, seek. That is the command that creates the sequence object. So I have to say seek, and then in the bracket, in inverted commas, I have to give the sequence as string. Here it is A, G, T, A, C, A followed by the other bases there. So it could be anything. Your sequence is given as a string, so it has to be in inverted commas. It is followed by a comma, and then you have to give the attribute of the sequence that we have. Here it is unambiguous DNA. So I say IUPAC dot unambiguous underscore DNA. So my seek is the sequence object that has been created with the sequence that I have given as string, as well as the IUPAC unambiguous DNA alphabet that I have given as an attribute. Now the sequence object has been created and it is stored in my underscore seek. If you want to display that my underscore seek, which is the sequence object, I just have to type the name of the sequence object here. So I just say my seek, my underscore seek, and seek. It displays the sequence objects. So the sequence in inverted commas as a string followed by the IUPAC alphabet, which is unambiguous DNA. So that is how we create and we display a sequence object. Sequence record objects. Sequence record objects are another type of objects, very useful objects, which can be used to record, store, as well as manipulate biological sequences. So earlier we saw sequence objects. It had only two attributes. Sequence record objects has many other attributes that we can associate with any given sequence. So seek record or seek record objects hold a sequence. This sequence is held as a sequence object with additional annotation including an identifier, a name, and several other descriptions. So the main and additional attributes of a sequence record object is listed below. The main attributes are ID, identifier, such as it could be a locus tag, it could be a string. So how exactly can I identify the sequence? That attribute is stored in ID. And then the actual sequence itself, that is stored in seq, seek. The sequence itself, which is held as a sequence object, or there are other type of similar objects which could be incorporated here, but we will just use it as a sequence object. So seek is a main attribute here, that in itself is a sequence object. So that is the difference we have to understand clearly. So earlier we saw sequence object. In sequence record object, that sequence object becomes one of the main attributes in sequence record object. Seek is that sequence record object. Additional attributes. The additional attributes are name, which contains the sequence name. It could be a gene name or it could be a protein name, and it is generally a string. And then the next additional attribute is the description. It could be some additional text which describes the sequence. So this is also contained as a string. And then it can contain attributes like database cross references. So it will be a list of databases. Once again, it will be a list of strings as far as the record object is concerned. And then any other features which could be recorded in feature attribute. So it will be sub-features defined, and it is a list of sequence feature objects. And then annotations for the sequence that we have. So this is further information about the whole sequence. It acts as a dictionary which could describe our sequence record object. We will see in the earlier slide we saw what is a sequence record object and what information it contains. In this slide we would see how to create a sequence record object and how to display that. We can create a sequence record object as follows. As usual, we will have to import the bioseq from seek. So I say from .bioseq import seek, and then we will have to import seek record bio.seek record from seek record. So I will have to say from bio.seek record import seek record. Here S and R in seek record are in caps. Python is case sensitive, so it has to be in caps to create the sequence record object properly. So we have to say from bio.seek record import seek record. So whatever I have given in caps should be in caps since Python is case sensitive. From on the next line, we have to say, as usual, we are handling alphabets, bioalphabets. So that also has to be imported from IUPAC. So from bio.alphabet, import IUPAC. So with all these modules being imported, we can create our sequence record object. As usual, I call my sequence record object as my underscore record. So we say my underscore record. The function that creates the sequence record object is seek record. So you have to say seek record with appropriate capital cases, which I have displayed here. So seek record. In the bracket, you can give the sequence. So seek is equal to seek, which we have already seen that seek with a capital S is function that creates the sequence objects. So I am creating a sequence object within sequence record function. So I say seek, and then I give the actual sequence. Here it is a protein sequence starting with the MKQ and it goes on. The sequence is given as a string and then comma followed by what alphabet, IUPAC alphabet it is. Here it is protein. I say IUPAC protein, close the bracket, and then give the attributes, main attributes and additional attributes one by one, which we saw in the earlier slide. So here I say ID is equal to the corresponding ID YP underscore 025.292.1. That is the ID for this particular sequence. So I say I give the ID and then I give a name for that. So name is HOKC, that is the name of the protein, and then a description. What is that protein? It is a toxic membrane protein, and the corresponding database cross references. Here it is empty, so I give an empty square bracket. So this command will be able to create a sequence record object in the name of my underscore record, and then we can simply display this my underscore record by saying print my underscore record. So this is the output that you will get if you say print my underscore record. So the ID followed by the name followed by the description, which is followed by the actual sequence object which contains the sequence as well as the attribute IUPAC alphabet. So we can also display individual attributes as follows. We say my underscore record dot ID and then several individual attributes could be displayed. So my underscore record dot ID displays the ID alone. If you want the name alone, we have to say my underscore record dot name. Manipulating sequences in a sequence object. So earlier we saw how to create sequence objects and sequence record objects. But most often in bioinformatics, we would read them from a file. We won't be creating sequence objects by typing them. But just to explain how it is created, I showed you to explain the structure of a sequence object and sequence record object. Most often what we do is we read the sequences from a file and then we want to manipulate those sequences. That is the bioinformatics task that we actually do. So here we will see how to manipulate sequences in a sequence object. Slicing a sequence is the most common manipulation that we do. So we will see how to slice a sequence from a sequence object. The same is applicable for sequence record object as well. So my underscore seek, I call it my underscore seek, which will store sequence object. So I am creating a sequence object first called my underscore seek and using seek, which we have seen already, with the following sequences which is displayed in the slide. It's an IUPAC unambiguous DNA. So sequence object has been created. It is stored in my underscore seek. If, for example, if I want to slice it from the 4th position to the 12th position, all I have to do is I will have to say my underscore seek and then give the index. So 4 is the index followed by a colon and then 12 is another index. So what we do is we get GATGGGCC. These are the alphabets, takes the indexes 4 to 12. Remember, Python starts with index of 0. So 0, 1, 2, 3, 4. 4 becomes G and it is followed, it goes on up to 12. So I have sliced the sequence and once again this creates another sequence object. It doesn't do a string manipulation. It slices the string and then puts it inside another sequence object. So if I display that, it will be seek, the sequence that has been sliced, and the alphabet information would be derived from the earlier sequence object that we have. So IUPAC unambiguous DNA. The new object which we have produced now, it retains the alphabet information of the parent sequence object. So earlier it was unambiguous DNA, and the new object also contains the unambiguous DNA. Now the other manipulation that we would like to do is turn the sequence object into strings. It is very simple. We just have to say string, and inside the bracket I have to give the name of my sequence object. Here it is my underscore seq. I just say string in brackets my underscore seq. For example, the following code constructs a simple FASTA format output. FASTA format is one of the most often used format in bioinformatics. So if I want my sequence object to be displayed in FASTA format, I just have to say FASTA format string. That is the name that we keep. So I have called it FASTA underscore format underscore string, and then I just give the first line, which is the greater than symbol, and then followed by the descriptions. That is a string, so it is in inverted commas, and then a percentage symbol followed by my seq. So I just have to print that, and it is printed in the FASTA format with the description my DNA sequence, which I have given in the command, followed by the actual DNA sequence. Concatenating or adding sequences. This is another important operation that we perform on sequences. So the actual concatenation can be done simply by giving a plus sign. So the concatenate operator here is plus, but we have to take care that we don't add two different sequences with different IUPAC alphabets. For example, in this slide you see I have created a sequence object called protein underscore seq, which is an IUPAC protein, and then another sequence object called DNA underscore seq, which is an unambiguous IUPAC DNA. If I try to add both of them, I say protein underscore seq plus DNA underscore seq, it will throw a type error, because here you are adding a protein sequence with a DNA sequence. That is not possible, and that is not necessary also. But if at all, very specific purpose, if you want to surely add both of these alphabets, then you will have to convert the alphabet information to generic alphabet, and then it is possible to add. So here, if I want to do that, in very rare cases, we just have to say from bio.alphabet import generic alphabet, and then you create your sequence object with generic alphabet as your ID, and then add both of them. So that is what has been explained in this slide. So in rare cases, we say protein underscore seq dot alphabet is generic alphabet. I am saying the alphabet here is generic alphabet. Similarly, for the DNA underscore seq also, the alphabet is generic alphabet. So we use the method add dot alphabet to convert the alphabets. So in Python, whenever we use a method, we follow it by a dot symbol. So DNA underscore seq dot alphabet is equal to generic underscore alphabet. When we do this, the alphabet information automatically gets converted to generic alphabet, and then we will be able to concatenate them. So protein, after this we say protein underscore seq plus DNA underscore seq, and then we get a new sequence object with a generic alphabet information. You see, it gives the sequence. The first few are protein sequences, followed by the alphabets of DNA sequence A, C, G, T, and the alphabet information here is empty. It doesn't contain anything because it is generic. We have concatenated them. Nucleotide sequences and reverse complements. So this is another important function that we will always try to do on sequences. This is one important manipulation that most of us do in DNA sequences. We try to get the reverse complements. So here in BioPython, it could be achieved very easily. So what I do here is, for nucleotide sequences, you can easily obtain the complement or reverse complement of a sequence object using the inbuilt methods of BioPython. So I import all the modules from bio.seq, import seq, from bio.alphabet, import IUPAC, and then I create a sequence object called my underscore seq, which we have seen already, and then the next line displays that my underscore seq, and then the complement, converting it to complement is very simple. I have already told you that any method Python should be followed by dot. Here we use the complement method. So first I give the sequence object's name, which is my underscore seq, and then the dot operator followed by a method called complement. If I want complement, I say complement, and then it should be followed by an empty bracket. So here you see the output, the given sequence, we have the complement given sequence and the alphabet information is retained. Similarly, if I want the reverse complement, I just have to say my underscore seq, which is the name I have given for my sequence object. So, my underscore seq dot reverse underscore complement with an empty bracket. We get the reverse complement as a sequence object and alphabet information is once again retained here. DNA sequence transcription. Transcribing a given DNA sequence is another important manipulation that we perform on sequences. So, what we do here is we have the coding strand. The seq object's built-in transcribe method is going to be used here. I am creating a sequence object by the name of coding underscore DNA. So, I say coding underscore DNA and then create the sequence object. The alphabet information I give is unambiguous DNA. So, I say IUBAC unambiguous DNA. And then I want my new transcribed sequence to be present in the sequence object with the name of messenger underscore RNA. So, that is the name that I select. So, I say messenger underscore RNA and I just have to say coding underscore DNA dot transcribe with an empty bracket. So, I can display, so all the new transcribed sequence is present in messenger underscore RNA. If I want to display that, I just have to type messenger underscore RNA and it is displayed as a sequence object. So, you can see that sequence and you can see all the T's being converted into U's because it is an RNA. So, ATG, the DNA coding strand started with ATG and you see that it has been converted to AUG here. And very importantly, you see that the alphabet information automatically gets converted to IUPAC unambiguous RNA. DNA sequence transcription and mRNA reverse transcription. The same way that we translated the DNA sequence, it is also possible to do transcription and reverse transcription. To do a transcription with a template strand from the above example, this becomes a two-step process. So, you say template underscore DNA dot reverse underscore complement with an empty bracket, which is followed by another method called transcribe. So, dot transcribe with an empty bracket. So, it creates a sequence object with the appropriate manipulation that we want to do. Here, so once again you see that the alphabet is converted to IUPAC unambiguous RNA. Similarly, an mRNA sequence can also be back transcribed to coding sequence as follows by using the dot back underscore transcribe function. So, messenger underscore RNA is the name that I keep. So, I am creating a new sequence object called messenger underscore RNA, which is going to contain the sequence object after the manipulation that I want to do. So, we have the messenger RNA, messenger underscore RNA is equal to seq, and then you have the actual RNA sequence with IUPAC unambiguous RNA as the alphabet information. So, that could be displayed. So, messenger underscore RNA, we have the sequence object of the messenger RNA now. Now, the manipulation that I want to do on this is back transcription. So, all I have to say is messenger underscore RNA dot back underscore transcribe with an empty brace. So, we have the new back transcribed sequences. So, here that is displayed and you see all the U's has been converted to T's. Similarly, the alphabet information is also converted to DNA. Translation from mRNA sequence. How to do a transcription? Similarly, a translation can also be done. So, both mRNA sequence and coding DNA sequence can be translated using translate function of the sequence object. So, we create the actual sequence object as described below and I have it in messenger underscore RNA. All I have to do is I have to say messenger underscore RNA dot translate. So, in our example, we have stop codons in that I have purposefully taken an example which contains an intrinsic stop codon. So, when it is converted to protein sequence, the appropriate positions where we have the stop codon is denoted by star, and then the sequence object has, it is an IUPAC protein, alright, that information is there, but it will also say has stop codon and it is described by star. So the sequence object will contain the actual sequence as a string with a star at appropriate stop codon positions, and the alphabet will also contain an information saying that it has stop codon. Translation and translation tables. So now in the earlier slide we saw how to perform the actual translation. So you can also translate directly from the coding DNA sequence. What we saw earlier was from coding mRNA sequence. So coding DNA sequence is, I have created a coding DNA sequence in the name of coding underscore DNA by the usual sequence object creation method which we have discussed already. So what I have to do now is, in order to translate that, I just have to say coding underscore DNA dot translate, and appropriately it is translated and the sequence object is created. So stop characters here also are denoted by star, which describes the stop codons. Most often what we want to do is we want to use a different codon table. There are several codon tables which the information of that can be obtained from NCBI. If I want to use something else, not the generic codon table, for example, I want to use a vertebrate mitochondrial codon table, then all I have to say is coding underscore DNA dot translate, and then in the bracket I have to say table is equal to vertebrate mitochondrial. That is a string, so it has to be in inverted commas. So vertebrate mitochondrial. So what now happens is the dot translate method, it takes information from the vertebrate mitochondrial codon table and then it translates accordingly. So you see here the resulting sequence object contains a different sequence than the one that we used earlier. So there we had two stop codons. Here it is proper with, it ends in a stop codon, so it ends in a star, so it doesn't have any intrinsic stop codon. It is also possible to specify the codon table in the dot translate method. So these different codon tables are given standard numbers in the convention. So this could be obtained from the gene bank file itself. So we say coding underscore DNA dot translate, and then I say table is equal to 2, which means that it has to use table number 2 for doing the translation. It has to use codon table number 2 for doing the translation. So codon table number 2 is associated with vertebrate mitochondrial codon. So you get the appropriate sequence object with this particular codon table. Parsing sequence data or reading sequence data in a more meaningful way from sequence files or directly accessing it from any sequence databases. So that's what we call parsing. Biological sequences are usually very long, and we might often have a single file that contains many such sequences. It is quite common that we encounter a big file, say for example in FASTA format, containing thousands of sequences. So our program quite often has to read these sequences, as well as it has to derive information from such a long sequence file. So BioPython has a bio.seq.io module which helps us to parse such kind of sequence data. So the BioPython function bio.seq.io.parse with an empty bracket is used to read in sequence data as sequence record objects, and this function expects two arguments. You have to provide two arguments to this function. The first argument is a handle, and this handle is used to read the data from or a file name. A handle is typically a file operand for reading, but could be the output from a command line program or data downloaded from the internet. So it could be anything. It could be an output from a command line program, or it could be some data downloaded from the internet. And the second argument that we will have to supply to a BioPython parser is a lowercase string specifying the sequence format. You will have to say in what format you need to parse your sequence data. So there is an optional argument alphabet to specify the alphabet to be used. This is useful for file formats like FASTA, where otherwise bio.seq.io by default will use a generic alphabet. Now, how do we read sequences from file? In general, bio.seekio.parse is used to read in sequence file as sequence record objects and is typically used for used with a for loop like what is shown in the slides here. So from bio import seekio, I introduce a for loop. So put a for loop, and inside the for loop I will have to give in my seek.io.parse function with the arguments that was explained earlier, which is the file name as well as the format in which I want to pass this data. And later on, since it creates a sequence record object, any of the attributes, main attributes as well as additional attributes, can be printed by using appropriate functions. For example, if I want to print the ID alone of the sequence record, all I have to say is print sequence underscore record dot ID, and I will have the ID. Similarly, if I want to print the sequence, all I have to say is sequence underscore record dot sequence. And we can also use additional functions along with this sequence record object. For example, if I want to know the length of the sequence, which is not present in which is not a data that is present in the sequence file itself. It is a derived data that we have to obtain from the sequence file. So what I have to say is print, in the bracket I have to use the len command, len, and then I will have to give the sequence record object. So let us see how we can read a sequence from GenBank file. GenBank file is a quite common file format that we encounter in bioinformatics. It is a file which contains the sequence as well as it is also rich in information. It contains many information. So how can we read this particular GenBank file and extract the information that we want? It has many, as I told before, it has many information, and we have to selectively take we might need selectively some of the information from the GenBank file, and we need to know how to extract that information alone from a GenBank file. So as I explained before, you have to import bio from seekio. So you have to say from bio import seekio, and then introduce a for loop, and then use seekio.parse module, given the arguments, which is once again the file name and the file format. Here we are going to use a GenBank file format, so we say GenBank, and then we will just have to print every record that I want. For example, if I want the length of the sequence record, all I have to say is print len seq underscore record. Parsing sequences through internet. So that is one important application that for which most of us write our codes. Most of the sequence information is huge, and they are present in databases in the internet. So we will have to access, quite often we will have to access these databases to get our sequences, to download our sequences. We can use BioPython, have the functionalities inbuilt in our code, so that when we execute the code, it automatically accesses the databases through the internet and takes the sequences and then manipulates them. You don't have to download it separately and then do the manipulation using your code. So this is an important, very useful BioPython functionality. For doing this, in this example, we are parsing GenBank records from the internet. So we can use Entrust eFetch interface, which will connect to the NCBI and get the sequences from GenBank using their GI numbers. So what should I do here? In order to perform this operation, I have to say from bio import Entrust, from bio import seekio. Seekio is the module that does the parsing. So I say seekio for sequence input and output. So from bio import seekio. And then it is important, while using Entrust, we will have to give our email ID. So I say entrust.email, and then you have to give your email ID there. So just to be generic, I say your email here, but you have to give your valid email ID in that place. So your email ID is given, and then we are creating a handle. So handle is equal to entrust.efetch, and then in brackets, what is the database that I want to access? It is a nucleotide database. So I say DB is equal to nucleotide, and then retrieval type I want it as FASTA sequence, retrieval mode is text, and then I give the GIID, which is 6273291, and that's all. Close the bracket and then enter. You automatically have the information in the handle. Now in order to pass it, I have to say seek underscore record, and then I use the seek IO module. So seek IO dot read, and then I give the handle here. I created a handle, and that handle is given as an input here. So handle, comma, I want it as FASTA, so I say FASTA. I close the handle, handle dot close. Now if I print that, it automatically prints as a sequence record object correspondingly. Here I print the length of the sequence, so I use the length function. So our output will be the GIID, which is what I print as a string by saying %s, and then gene bank accession, and then all other parameters that I want to print. So we have parsed using /if/h, we have connected to gene bank, and we have passed the sequence of the given sequence ID. Parsing Swiss-prot sequences through internet. In the earlier slide, we saw how to parse a gene bank sequence. Similarly, it is also possible to access other databases. Here, this is another example where we parse a Swiss-prot sequence database. So here, what do? I once again use bio.seek.io and then the read function of that, and then I'll have to import the XPASI module. That is where we get the data from. So I say from bio import XPASI. Cases should be caps as appropriately as it is shown in the slide. So E caps and PAS caps. And then the next line I say from bio import seek.io. We need the sequence input output module. So I say bio import seek.io. Give the handle. So XPASI dot get underscore Swiss-prot. So I say sprot underscore raw and give the sequence ID. Everything else is the same as what we saw earlier for GenBank. So we have created a sequence record directly by accessing Swiss-prot, and we have got the sequence of the given ID. So now if I want to print that, first I say print sequence record.id, it prints the ID, and then record name, I just say seek underscore record.name, and everything, the description. If I want the description, I say dot description. Similarly, all other attributes can be printed. So the output of the corresponding code is given on the other side. So you get all the information. So you get the ID, the record name, the enzyme commission number, or whatever data that is stored in the actual sequence, and then the length of the sequence. I have used the length function len, so the length of the sequence, and the annotations also. So the annotation says it is an acyl transferase, it is involved in flavinol biosynthesis, whatever data that is stored additionally along with the sequence and the annotation attribute, that is also displayed here. So in this slide we saw how to parse a Swiss-prot entry using BioPython. Converting between sequence file formats. This is the most often used method. In bioinformatics, we always want to convert sequences to different file formats based on what program we are going to use. BioPython has very useful and very simple modules to perform this task. So here, for example, if I want to convert a gene bank format file which is named as test.gbk, and then I want to convert that to FASTA, it is very simple. I just say from bio import the sequence IO module. We have seen that already, and then it is stored in records. I have stored that in records sequence IO parse. The actual file name test.gbk that contains the gene bank sequence in gene bank format, and then I give the format, it is gene bank, so it is stored in record, and then I say count, that is the name I select, sequence IO dot, I write the records in my underscore example dot FASTA in FASTA format, and then I can just print them the usual Python convention. I just say print converted sequences. So this will be able to convert the gene bank sequence into FASTA sequence and then print that. We can also have a helper function in BioPython to do the same. We just say, to use that function, we just say, we have to input the sequence IO module. So from bio import the sequence IO, and count is the name that I have created. So I say count sequence IO dot convert. That is the method that does the conversion. So I say seek IO dot convert, and then give the file name. I want to convert GenBank file into FASTA file. So name of the GenBank file is test.gbk and the format is GenBank. So I give them both first, followed by what destination do I want. I want the data to go into a file named my_example.fasta. Give that file name there, and then what format do I want to convert it to. It is FASTA. So I say FASTA there. So all these 4 parameters are necessary for .convert function. So now the sequence automatically gets converted from GenBank to FASTA format, and that could be printed as well using the usual Python methods. Parsing sequence alignments. As we saw earlier, we pass sequence data in a meaningful way. In the same way, it is possible to parse sequence alignments as well. Handling sequence alignment in normal programming is very difficult. You will have to, the sequence alignments are of different formats, as well as it is much more complicated than simple sequences. We will have to first read the format, understand the alignment, and then we will have to do our manipulation with the alignment. Whereas if we use BioPython for that, it all becomes very simple. We have a bio.align.io method that could read as well as parse sequence alignments, and it helps us to manipulate as we manipulated the sequence record objects which we saw earlier. So sequence alignments can be read using bio.align.io.read or bio.align.io.parse. So it's the same as sequence IO. Instead of sequence IO, we have align IO. So bio.align IO. So using bio.align.io is also the same as how we use the sequence IO function. So what we have to do is we need a regular iterator. So we can use a for loop, a regular Python for loop, inside which we can use our bio.align.io.parse. So it gives multiple sequence alignment objects which can be used in a for loop. Bio.align.io.read function returns a single multiple sequence alignment object. Both functions expect 2 mandatory arguments. The first argument, as usual, like that of a sequence IO, is a handle to read the data from. So you will have to specify from where you are going to read the data. The second argument is a lowercase string specifying the alignment format. What format do we have in an alignment? In the earlier slides, we saw how we can parse sequences, biological sequences. In the same way, BioPython has functionalities to parse sequence alignments as well. So parsing sequence alignment. Consider the following alignment. This alignment, even though it is difficult to read, is very rich in information. This format is called as a Stockholm file format for sequence alignment. So it contains all information about the alignment, but it is not easy for visual interpretation. Suppose if I want to manipulate this alignment or convert this alignment with rich information to some other form of alignment, it is also possible to perform that task in BioPython in a very simple way. So in the earlier slide we saw about a Stockholm file format. Now in this slide we will see how that Stockholm file format can be meaningfully displayed so that there is a clear visual interpretation. To do this parsing, what I do, I have to import align.io method from BioPython. So from bio import align.io. Align.io module has been imported. Now alignment is equal to align.io.read, give the name and .sth file. It is a file name in which the alignment is present, and then it is in Stockholm format. So I say Stockholm. That's all. This .read, align.io.read function will be able to take all the data and it will be able to put it in a meaningful format. So visually easily interpretable format. So we will be able to display that. I say print alignment. Alignment is the name that I have given to create this. So I say print alignment, and this will be the corresponding output. Only a section of that is shown. 7 rows and 52 columns are shown here. This is more easier for visual interpretation. You have the actual alignment, multiple sequence alignment, followed by IDs or descriptions about the sequences. Converting the formats of alignments. As we converted different sequence formats, it is also possible to convert alignment formats. So in this example, we see how to convert a PFAM or Stockholm file format to ClustalW file format. So I have created an alignment in Stockholm file format. So we just say count align IO dot convert. That is the method that does this conversion. So I say align IO dot convert, give the file name followed by the actual format, which is followed by the file name in which I want to store the new alignment and its format. So here it is Clustal, so I say Clustal. That's all. It will be created and it will be stored in count, and this could be printed by using the usual Python methods. So I say print command, and then the output will be as shown in the figure, which is the Clustal format. So Clustal X format. It is also possible to manipulate alignments. In the earlier slide, we saw how to convert different alignments into different formats. It is also possible to manipulate the alignments. It is very similar to how we manipulated the sequences, how we sliced the sequences. For example, if I only want the rows from 3 to 7, the sequences from 3 to 7 of an alignment, all I have to say is print alignment in an empty bracket. I say 3 colon 7. So it displays third row to the seventh row. So third sequence, fourth sequence, fifth sequence, sixth sequence. In Python, the slicing is always done before the index that is given. So it starts with the index 3, third row, fourth, fifth, sixth, and then it cuts before seventh. So you will have only 4 rows and the corresponding sequences and alignments. So that is important. This is BioPython, and usual Python conventions are followed here. Slicing is always done from the index and to before the index. All slicing ends before the index that is given. That should be remembered. That is the usual convention in Python, and that is followed in BioPython as well. So if I say 3 to 7, 3 will be included, but 7 will not be included. It stops before 7. It stops at 6. So that is important, and that has to be remembered while manipulating alignments. Here in the earlier slide, we manipulated the alignments, we sliced the alignments based on rows. It is also possible to slice the alignment based on columns. So suppose if I want to print the alignment from 3 to 6, and then I want the first 6 columns. All I have to say is print alignments 3 colon 6 comma colon. I want the first 6 columns. So the descriptor for the first can be left empty. By default means from first. So from 0, that is from the first, and then I leave it empty. I want the 6 columns, so I just say 6. So this print statement displays from rows 3 to 5 followed by columns 0 to 5. So it is also possible where 2 blocks of alignments can be added. So we have 2 different alignments and they can be added using BioPython. But we have to notice that we have removed the gap regions in the example that we have shown there. So if I want to remove the gap regions, I can just edit the alignment with appropriate column numbers and then add them together. That will remove the gap regions. So in our earlier slide, we saw some gaps that can be removed in this particular example as shown here. So the edited sequence, I store it in edited, and then I say alignment, give the appropriate row numbers. Here you see I have given an empty colon, which means all the rows starting from 0 to the end. By default, if I just give, you don't have to give any numbers there. If you want, you can give, but it is simpler. You don't have to give any numbers. If you give just a colon, by default, Python interprets that as from the beginning to the end. So the same way as we specified the columns there. In the earlier example, we specified the columns with an empty colon and 6 from the start to 6. Similarly, an empty colon here says from the beginning to the end, all the rows followed by the first 6 columns. So from the beginning to column number 6. So comma colon 6. That edited piece of alignment is added from alignment all the rows once again, followed by 9th column to the end. So 9 colon and end, and that could be printed appropriately. Running BLAST using BioPython. BLAST, basic local alignment search tool, that is an application that is used very often in bioinformatics, that could be run directly in a BioPython code. It could be inbuilt in your BioPython code. So how to do that? So to search against the nucleotide database NT using BLAST-N, when you know the GI number of the query sequence, all I have to say is from bio.blast, that is a module. So bio.blast import ncbi www, and then I create a result handle and say ncbi www.qblast, and in the bracket I give the information. I want to use the BLAST-N program, so I say BLAST-N, and the database that I want to search NT and the GI number. So automatically it connects to the internet, does the BLAST search, and the results are stored in result underscore handle. So the query sequence from a FASTA formatted file, for example, if you don't want to give the ID, you have the file in your system, it is also possible. All you have to say is, you have to open the file, so test.fasta here, and then you have to read that, and it is stored in FASTA underscore string. That information is given in ncbi www.qblast. So if you already have sequence object for the FASTA file, it is also possible to give that sequence object in while performing BLAST. All you have to say is mention the BLAST program that you want, the nucleotide base NT, and then the record.seq object of your sequence record. Passing BLAST output. So that is also an important task. So when we perform BLAST search, which we quite often do in bioinformatics, almost most of the 90% of the bioinformatics task, sequence manipulation or sequence analysis task that we do starts with performing a BLAST search. So BLAST output will contain numerous alignments. It might contain numerous alignments depending on the sequence that you are analyzing, and all of the alignments that we see need not be meaningful. So only few of the alignments will have a meaningful statistical score behind them to be considered as a significant alignment. So it is very important to parse a BLAST output and take the data that we require, only those data that we require. So an XML output is more stable and is much easier to parse. So what we can do is, we can have the BLAST output in an XML format, and then that could be used in BioPython, which is much easier, which becomes much easier to parse the data. So for example, we just have to say from bio.blast import ncbi xml, all in caps. So now we just have to say blast underscore record somewhere where we have to blast underscore record is the record where I am storing my results. So I say blast underscore record is equal to ncbi xml dot read, and then I'll have to give the result handle. So that helps me to parse blast output from the XML format. BioPython has very useful, simple methods for parsing blast output, and all we have to say is to, for example, from a blast output I want to select sequences that have a better e-value of better than 0.04 from all the results that we have obtained after a blast search. All I have to say is put that as a threshold. So I say e-value underscore thresh is 0.04, and then two loop constructs of Python for alignment in what? In the blast.alignment underscore records.alignment, and then for HSP in alignment.hsps, then if hsp.expect, that is the method that looks for the expectation value, that is e-value, is lesser than the threshold I have set. So that goes inside the if construct, and then I can print whatever attribute of blast result that we want. If you want the alignment, you say print alignment. If you want only the sequence, you say print sequence. And if you want the alignment title, give it there, or e-value alone. It's possible to print any of the alignment attributes that is below this e-value threshold that I have set. So it becomes very simple. Even if your blast search brings in thousands of results, you can put appropriate filters in your code which can select certain alignments from the huge results. The PDB module for structural bioinformatics. So it is also possible to meaningfully interpret structured data and to manipulate structured data or to derive information from structured data using BioPython. The module for that is bio.pdb. So bio.pdb to work with structures. Bio.pdb has a PDB parser class that produces a structure object, and the structure object contains all the attributes that we need to access. So from the structure object, any data about structures could be retrieved. So in order to parse a PDB file, we create a PDB parser object first. How do we create that? We say from bio.pdb parser import PDB parser, and then I store it in P. So I say P is equal to PDB parser and permissive is equal to 1. And then I say structure ID, I give the structure ID there. 1FAT is the structure ID. So I say 1FAT. The file name is pdb1fat.ent. So now we have the structure. So I call it yes, it could be anything. So I say yes is equal to p.get underscore structure bracket structure ID and the file name. Now the permissive flag is to ignore. I say permissive is equal to 1. That flag ignores the common problems associated with PDB entries. Sometimes there could be missing residues in PDB and there could be multiple positions. So the common problems that are associated with PDB entries are ignored by setting the permissive flag as 1. Structure representation. So here we saw how to import structure data and create a structure object in the earlier slide. Now we will try to understand how a structure is represented in a structure object. So SMCRA architecture is the architecture by which PDB structure is represented in a structure object. So what is that SMCRA architecture? Structure, model, residue, atom architecture. So that is the hierarchy. So a structure contains models, a model contains residues, residue contains atoms. So if you want to access this information, you have to create for loop constructs appropriately in that hierarchy to obtain information. For example, if I want the atom information to be printed, I have to use PDB parser, get the structure, and then I have to put the loop constructs. So that is what is explained here. So I say P is equal to PDB parser, and then structure is equal to P.get. So the PDB parser is in P. So I say P.get underscore structure and X, whatever structure that you want, and then appropriate file name. So the structure object is present in structure. So for model in structure, it iterates over the models present in structure. If it is an NMR structure, we will have several models. Crystallographic structures have only one model, so it will just iterate once. If it is NMR structure, it will iterate over as many models a structure object contains. So for model in structure, and after the model it goes to chain. So for chain in a model, so a single model can have more than one chain. So for a chain in model, and then it goes to the residue. For every residue in chain, and then it goes to the atom. For every atom in the residue, we print atom. So that is the hierarchy that we always have to maintain. So that prints information about the atoms in any given structure. Retrieving information from structures. For loop constructs that I have explained in the previous slide. Here we will see how to select coordinates of all the C alpha atoms in a structure with B factor greater than 50. So I use the same loop models, loop constructs for loops. So for model in structure, get underscore list. For chain in model, get underscore list. So it lists every chain in that model, and for each of the chain, for residues in each of the chain. So I say for residue in chain dot get underscore list. If the residue has underscore ID CA, if it has a CA atom, then Ca is equal to residue Ca, and then I put the if construct. If Ca dot get underscore B factor is the method that gets the B factor of that particular C alpha atom. So I say get underscore B factor, it gets the B factor and then checks whether it is greater than 50. So I say greater than 50 followed by colon. That is a usual Python convention. If statements, for statements should be followed by a colon, and then if there is Ca atom which has a B factor of greater than 50, then it goes into the if construct and then prints that particular Ca coordinates. So the coordinates can be obtained by Ca dot get underscore coordinate with an empty bracket. So earlier we saw how to selectively get information from structure object. Now we will see how to analyze structures. So analyzing structures. For example, if I want to measure distances between two given C alpha atoms, I use the minus operator. It is very simple. So I just say Ca1, that is atom number 1, is residue 1 of whatever the atom description that I give, Ca. And then Ca2, another atom that to which I want to measure the distance, that is given there as information. The distance can simply be obtained by distance is equal to Ca1 minus Ca2. That distance is stored in distance and it could be printed. Similarly, angles can also be very easily measured in BioPython using calc underscore angle function. So I say vector 1, atom 1 dot get underscore vector. So it gets the information as a vector, and then vector 2, second atom, vector 3, third atom. Vector 3 is equal to atom 3 dot get underscore vector. An angle can be calculated very simply. I say angle is equal to calc underscore angle, give all the three vectors that we created earlier. So all the three atoms, corresponding angle is stored in angle that could be printed. So the variable angle that we have created will contain the angle information between these three atoms. Torsion angle can also be calculated in the same way, but we have to give 4 vectors here describing the 4 atoms that for which we want to calculate the torsion angle. The function is calc underscore dihedral. So students, let's now summarize what we have learnt in this module. This tutorial was aimed at providing a fundamental knowledge about BioPython methods for handling sequence and structure data. BioPython have different functionalities to pass bioinformatics files into Python utilizable data structures. Support for biological data in different format is also available in BioPython. It also deals with popular online bioinformatics portals like NCBI and XPSI. Interfaces to common bioinformatics programs such as BLAST, ClustalW and Emboss tools were also explained in this tutorial. Thank you.